So what you see here is a transcription factor, AP1. And as you know, transfac is a database about transcription factors and their binding sites. And now we're dealing with these very important proteins, which are living and functioning in the nucleus of the cell. They are binding to the DNA in a specific manner, so in the special places which the transcription factors can recognize. We call them binding sites. And due that binding, these transcription factors can actually kind of anchor at the place. And then by the activation domains or other domains of those factors, they can interact with other factors, they can interact with transcription machinery, and they can regulate expression of the genes. And the transfac, a big part of it is actually concerned with recognition of binding sites in any sequence. And why do we need to do the prediction, the recognition of binding sites? Because so far we still very far away from knowing all functional binding sites in genome. And I talked about that before. So we have so many transcription factors in various cells which are functioning in various conditions, and they can bind to their consensus sequences in DNA. But we have a number of technologies, experimental technologies, to understand where they are binding, to identify those binding sites experimentally. And now we have high throughput technologies like ChIP-seq or TAC-seq, which allows us to really find many binding sites for a factor in the whole genome. But still, our simple estimates telling us that still we are very far from knowing every possible sites in genome. Maybe we know just less than 1%, or even maybe 0.1% of all binding sites in genome. So we still need to predict. And how we do that in transfac, as well as in other databases, which you can see here, we basically collect so-called positional weight matrices, and you see them. One example of that matrix here in the screen. It's a positional weight matrix for a factor which is called AP1, and that was the picture I presented in the first slide. And this positional weight matrix is built from experimentally known binding sites, which we collect in transfac, or we grab them from experiments, high throughput experiments, like many other do in other databases. And then we build, we basically count how many times a particular nucleotide was found in a particular position on that site. And then you see those cases when almost in all cases T is staying here in the first place. Then this letters is big in that log plot. And if the position is kind of redundant, so it can be several nucleotides staying here, then those letters are small. So they not only present the motif, which this factor recognizing in DNA, but also presenting kind of a conservativity of particular positions in that motif. And as you see that transfac is actually, we're making our efforts in transfac to collect as many as possible those positional weight matrices by integrating knowledge from many existing databases, from literature, which we are scanning, and by building these matrices by ourselves. So, and the whole idea of those matrices is actually to find new binding sites in genome. As I said, we still have to find them. So, because we don't know where those factors can bind, and if we don't know, we cannot actually explain how factors regulate the genes, regulate the gene expression. And but still, let me go a bit, in details how this all prediction works, because that's still a more often very frequent question. to understand how we predict those sites and whether our predictions are, first of all, whether they are correct and how they correspond to real affinity of those factors to their targets. Okay, so let's go step by step. So first, as I said, we have a collection of known binding sites, which I aligned, like here. You have, for instance, 5 sites, which are experimentally known for a given factor. And we align them to each other according to the most conserved places in that site using various algorithms. And then we count these positions. And from that counts, we can build a matrix which is called a count matrix. Basically, we have number of cases observed here, and then the first transformation we do, we basically normalize these counts by the number of sequences which were observed in that position, and then you have here the probability matrix and position specific frequency matrix, PFM, you can call it. So it's a frequency of a particular nucleotide in particular position. But then a certain transformation is useful, so-called the log score. So what happens here, in fact, as you may understand, that these probabilities which we have here are difficult to tackle, sometimes algorithmically, because if you want to find a new sequence which fits to that matrix, you basically have a sequence, and then you go through that matrix and you have to multiply these probabilities in order to get the probability of that sequence to appear. Why do we need to know probability? Because if we see that the observed sequence have a very high probability to be the binding sequence for that factor, we can claim that the sequence we found is a true binding site. But again, by multiplying those numbers, and especially when you have a long motif, or you have such places where you didn't observe anything, so zeros, they can really distort your algorithms, and that's why the useful thing is here to make a logarithm of those values and to use so-called pseudo counts. So as you can see here, we don't have any irrelevant values which could happen if you have a logarithm of 0, because we add a small pseudo counts here, which kind of compensate our little knowledge about binding sites. So basically, we have our set of sequences, set of sites, and it's a limited set. It's not unlimited. So, for instance, only 5 sites, and our observation is, number of observations is very small. So we have to compensate the small number of observation by a certain pseudo counts. So that's why we got this logarithmic values, and then these values can be used further to estimate the score of the given sequence, and then by simply summing up those values when you see a sequence. So you take this matrix, you put it on the sequence, and then you simply sum up those values to each other, and you got a certain value. We call it a score, a log score. And if this value is high, then basically we can say that this sequence is very similar to the binding sites of transcription factor, of a given transcription factor. And another useful thing for the logots is actually, if they are negative, it means these values are very small, and they contribute, in a negative side. And basically, if you have a sort of score near 0, you may say that this site is nearly random. So okay, so that's how we calculate these logout scores and predict binding sites. By the way, in our actually more advanced algorithm, which we call match, we use additional weighting, which is weighting those positions in addition by a certain information content. So it's like a double weighting, telling that a certain position is really important, it has a high information, and we give additional weight to those positions. But that's a special case, and now I talked about that in my previous talks. But here, more traditional and way, which is also applied in other algorithms, which is called log odds, when you simply sum up different sum up position weights of consequent positions. And then we made some efforts several years ago, and it was published in this paper by Stegmaier and et al. In PLOS 1, where we basically generated a lot of random sequences and estimated probabilities so that a given score can be observed by a random chance. So we basically we calculated a p-value, so this famous p-value. So it's not enough to see just the score, but you have to say how probable to observe that score by random chance. And why? Because if it's observed and it's very rare, so p-value is 1 in -10, for instance, you can say it's a rare event, and that's why this must be a real binding site for a transcription factor. So we computed the score for many random sequences, and then we even made our estimates based on actually on the curves. Like here we used a mixture probability model to estimate those p-values, which allows us actually to do these estimations not by random generating random sequences each time, but by just estimating certain parameters of those distributions, and then estimating those probabilities. So why again? So we want to know if we observe this score, how probable to have this score for that particular matrix by random chance. So, and now actually what I want to talk today, and I will show this in online in our system, that we not only compute this probability for a given position of a transcription factor to bind to that position, so basically probability of that binding sites to be a true binding site, but we can try to estimate a sort of affinity of that transcription factor to the whole region. Why should we do that? Because we never know how the true regulation of that particular gene is happening. So we know that if this sequence is, for instance, a promoter of a given gene, which is regulated by AP1 transcription factor, then in that promoter we can observe several binding sites for that one transcription factor. And some of those binding sites are very strong, so which means they have very high log score. So actually very low p-value, which means they are very improbable by random chance. So they are very good candidates for binding for those transcription factors. But there are a number of other positions in that promoter which have low scores, so lower than this maximum, but they still can be targeted by this AP1 factor. Why? And I was actually discussing that last time, because in many cases, transcription factors, they interact with their target sequences in the complexes with other factors. And for instance, there are many cases when these AP1 binding sites are occupied by AP1 factors, because there are other transcription factors recruit this AP1 to that place. So it's not really very high affin to that particular place, but it's compensated by, its binding to that place is compensated by the synergy with other factors. But all in all, this promoter is regulated by AP1. It means that many sites in that promoter can contribute to the whole affinity of that promoter to that particular transcription factor. It could be either by one place, which is a very high scoring, or it can be a multiple low scoring sites which are distributed over the whole promoter. But in total, they contribute the same amount of affinity to the target, to that transcription factor, to bind to that promoter, and consequently to regulate that gene. So that's why what we came up with here and published in that paper is a so-called affinity score, which is given to a whole promoter. So not just to one binding site, but to the whole promoter or whole regulatory region, which we want to analyze for a given transcription factor. I'm sorry. So, and this is a formula how we calculate it. Basically, not going into many details of that formula, it's very simple. So we have in each position of that promoter, for a given matrix, we can calculate the log score. And that log score is actually exponentiated first, and then it's summed up through the whole length of that promoter. So it's actually basically more or less the the sum of the weights over the whole promoter divided by the length of that promoter. Well, minus the length of the window of that binding site itself. So it's a kind of average affinity to that promoter for that transcription factor. And the good thing here is that this score is in fact not depending on the cutoffs of individual cutoffs of that particular positional weight matrix. So because if we would do the standard motif search, we would first run this log score through the whole promoter, and then we have to cut this score by a certain cutoff, and then we kind of have to choose which cutoff to use, and that's always a question. We don't know what affinity, with real affinity, which factor is binding. And then if we select a high cutoff, for instance, somewhere here, we will find just one site, this one. If you select a low cutoff, maybe we find more sites, but then we find a lot of sites. So you see a lot of variability of your findings depending on which cutoff you choose. In the case of affinity score, we don't put any cutoffs on the position weight matrix. We calculate this log through the whole promoter, then we sum up the whole weight of those scores, and we have one number, one figure for the whole promoter. And of course, we can apply a new cutoff for that figure, a new probability for that figure, and that's something what we did, in our computations. What we had a question basically: if you have a random sequence, not a real promoter, but just a random sequence, and you apply this AP1 matrix to a random sequence, of course, you will find some matches to that motif, and maybe in some cases quite high matches, high scoring matches, and you would get this affinity score for random sequence. But if you have a real promoter which is regulated by AP1, we expect that this affinity score is higher than the average random sequence, right? So that's why we have to estimate how these scores are distributed randomly, and also make another efforts to estimate probabilities and p-values for those affinity scores. And what we did, and that's one example on one matrix, which is MyID. And actually, here is this affinity score, which is can be positive or negative, because it's average affinity basically over the whole promoter. And it has this multi-modal distribution, which is very interesting because actually, which tells us that, and that's very clear why, actually, because in many cases, these matrices have very few, very conserved sites, very conserved nucleotides. So the matrix can be of the length, say, 10, 12, 15, but maybe only 3, 4, or 5 nucleotides are really conserved. Like in these cases, we can say these 2 T and this A is most conserved ones. And maybe this one as well, the letter A. So which means if this particular site will be missing this particular very conserved nucleotide, you will get a very big jump in that score. So which means these high scoring sites have all 3 conserved nucleotides are really preserved in that site. This one missing 1 of them, this one missing 2 of them, and this one's maybe missing all of them. That's why their scores also so low. So, and then we when we basically sum up these distributions, we can also make a parametric estimation of the probabilities, and through that we can estimate p-values of that affinity scores. So that's a little bit of theory today, just to tell you that that's what we are calculating. Those affinity scores, they're not just randomly chosen, but we made a very important and basic computations to understand how they work. And there are a number of other matrices I show here how affinity scores are computed.